Follow us on social media and please hit the subscribe button so you don't miss out on any updates from our ALS experts.
Welcome, everyone. Every 2 weeks, your extended family shows up, and it's you who makes a difference. Bring that piece of that puzzle that you got inside of you, something you learned in the past 2 weeks, maybe. Whatever you bring with a smile. And if you don't have a smile on today, I want to give one to you. Thank you. Pleasure for you to show up. And with that, Lisa, it's all yours.
Welcome, everyone, and thanks so much for being here tonight for our Everything ALS Wednesday webinar series. We are so excited to bring experts right directly to your living room. So I'm Lisa Deegan. For those who don't know me, I'm part of the Everything ALS team. My younger brother had ALS, and each member of our Everything ALS team has personally been affected by ALS. So our goal is really to provide you all with what we didn't have while we were going through our journeys. So tonight, we're going to look forward to an evening in education on the latest research from one of the top experts in the field, Dr. Michael Benatar from the University of Miami, to present on symptomatic and pre-symptomatic biomarkers for ALS therapy development. After the presentation is finished, we're going to open up to questions and answers, and followed by a non-recorded private session where you could meet others face-to-face over Zoom, but connect with other people within our community, share your thoughts, tips, and tricks. And it's just a nice way to get to know others within the community that are perhaps going through what you're going through. So before we get started, please, one thing I want to mention is please consider signing up for our Voice Project to help end ALS. We're conducting the largest digital biomarker voice study that aims to get an earlier diagnosis of ALS and a better understanding of the course of the disease. So with the help of everyone, we will be able to discover and validate digital biomarkers that will help improve the success rate of clinical trials, measure effectiveness of therapies, and therefore help to accelerate treatment. So please visit everythingals.org/research to learn more about this and sign up. It's only 5 to 10 minutes a week, and all you need to be is 18 and older. So now for tonight's presenter, Dr. Michael Benatar. He is the Walter Bradley Chair in ALS Research, Executive Director of the ALS Center, Chief of Neuromuscular Division, and Vice Chair for Clinical and Translational Research in the Department of Neurology at the University of Miami. Dr. Benatar leads the Pre-Symptomatic Familial ALS Study, the CReATe Consortium, which focuses on therapy development for ALS, and he's also led both phases 2 and 3 in ALS clinical trials. He's internationally known for his pioneering work in defining the field of pre-symptomatic ALS, including the discovery of the first biomarker of pre-symptomatic disease that has been critical to the design and initiation of the first pre-symptomatic ALS trial. So Dr. Benatar has also been a thought leader in challenging existing paradigms for preclinical therapeutic studies, shaping how we use biomarkers for therapy development. He is passionate that all ALS patients have opportunities to participate in and contribute to research, so he has integrated research and clinical care at the University of Miami. He's also co-developed the ALS toolkit in the electronic health record as an instrument to help facilitate collection of research quality data at the point of clinical care. So now please join me in welcoming Dr. Benatar. We really appreciate all the work you do for ALS and your dedication of sharing it with our community tonight. So welcome, thank you.
Great, thanks so much, Lisa. I hope everyone can see the screen and the volume's okay.
Yes, perfect.
Yes.
Okay, good. Well, really appreciate the opportunity to talk to you this evening. And I wasn't aware, I was interested in your introductory remarks, Lisa. I wasn't aware that you were engaged in a digital biomarker project. And so this is perfect. I feel like I'm preaching to the converted. And so I apologize if I'm telling you things that you know already. But I do think that biomarkers are one of those things that we talk a lot about in the community, in which sometimes perhaps are misunderstood. And so what I'd like to do is spend some time talking about them, and then talk a little bit about how I think they have a role to play in therapy development, both as we think about people affected with ALS, that's the symptomatic piece, but also pre-symptomatic people who might be at risk for ALS, because increasingly we're trying to think about and move towards preventing ALS, not just treating it. So let's see here. I should say that I do have some disclosures. I do some consulting work for industry that are developing therapeutics for ALS. Both Orphazyme, a Danish biotech, and Biogen, based in the US, have licensed some of our research data for therapy development, and I serve as a site investigator on industry trials, most recently sponsored by both Orphazyme and Biogen. So here's what I'd like to do. I'm going to start by giving you sort of an introduction and an overview of biomarkers. I'll then, and that'll be about a third of what I have to say, I think, and then I'll talk a little bit about their role in therapy development for people with ALS, and then last but not least, we'll talk about pre-symptomatic disease. Okay, so we have lots and lots and lots of biomarker candidates in the ALS world. We're not short of candidates. And I've grouped them here in this table into three broad categories. Those that are biofluid based, so biomarkers that we can measure in biological fluids, blood, urine, spinal fluid. People have also done a lot of work using MRI and advanced imaging techniques to try to quantify the disease in ALS and to use imaging as a biomarker tool. And then broadly speaking, we have these neurophysiological or electrophysiological tools which try to make recordings from nerve and muscle to tell us something about the disease. And so notwithstanding this plethora of candidates, until very recently, I would argue, we really have not had validated biomarkers that are relevant to therapy development. And I think that is beginning to change. I think it has begun to change, but I think it's helpful to reflect on why it is that we've had all of this richness of candidates, but they really haven't moved forward to help us with therapy development. And I think there are a few reasons. The first is that we have to differentiate in our minds discovery of biomarkers, finding something new, exciting, interesting, and then the heavy, often grunt work of validating that. And validation is a loaded word. I'm going to come and talk about that some more, but it involves many different things. And I think validation often is a word that means different things to different people. So we'll come back and talk more about that. But discovering a biomarker isn't the same as validating it and figuring out how it can be useful for therapy development. But I think another reason why we haven't moved some of these biomarker candidates over sort of the finish line is that I think there's been a failure to appreciate the nuances of developing biomarkers that have what the FDA refers to as a well-defined context of use. And that's a funny term. What it really means is that biomarkers need to be fit for purpose. How do we plan to use them? What's their specific indication utility? And then the biomarker that has a different utility needs to have different characteristics and needs to do different things. And I'm going to dwell on each of these, but just to introduce the ideas, broadly speaking, we think of diagnostic biomarkers, prognostic biomarkers, predictive biomarkers. Those sound a little bit the same, but I'll clarify their difference. And then perhaps the holy grail, these pharmacodynamic biomarkers, biomarkers that show us that there's been a treatment effect. And I think the important thing, and I've said this, but I'm going to say it again, is that what we want of these biomarkers, so their performance characteristics, what we need them to do, really differs depending on their intended use. And I think there's been a large scale, perhaps underappreciation of the importance of that. And I'm going to spend some time trying to illustrate that for you. But before I do, I wanted to just sort of take a side path for a moment and talk about what the biomarker development process looks like. And I see this broadly as falling into four categories. There's discovery. There's what one might call analytic validation. There's then exploring the clinical utility of the biomarker. And then perhaps there's this process of qualification, which means going to the FDA and trying to get that biomarker approved to use in a certain way for therapy development. So what does discovery mean? Discovery is establishing proof of concept. It's an early idea. Very often when this is being done, scientists are using clinical samples of convenience, what they happen to have stashed away in the freezer. And they're beginning to think about, hopefully, although often not, what would be the use of this? How would it be used in therapy development or in clinical practice? We then get into a phase of analytic validation. And what this means is we have to figure out, how does this biomarker test actually perform? Does it matter how we measure it? Does it matter what temperature the experiment is done at? Does it matter if the sample has been frozen and thawed? How many times? Does it matter if the sample sat on the bench after it was collected before it was frozen? Does it matter if the reagent of the antibody was from this lot, from this laboratory, or a different lot from a different manufacturer? So we have to figure out all of those details. We have to understand the impact of potential confounding factors. Does it matter if we measure the biomarker in the morning or at night? Does it matter if the person was fasting or not fasting? What about the effects of medications? What about the effects of coexisting diseases? We need to understand all of those things, and we need to begin to refine that context of use. Once we've done this analytic validation, we start to get into the clinical utility. Here we're looking at large, carefully defined clinical cohorts. These are often clinical trial-like cohorts. We need very well-defined SOPs, standard operating procedures. How do we collect, for example, a biological sample? How do we process it? How do we store it? How do we do an electrophysiological technique? How do we gather that image on the MRI scanner? What kind of scanner? What software on the scanner? The magnet in the scanner may perform differently in the morning or the evening. We have to refine and sort out all of those details. And we really have to establish how we expect to use this biomarker, either in clinical practice or in a research context. And then very little goes beyond this, but if we do, we're getting into qualification with the FDA. And here we need to demonstrate the clinical utility. We need to write down what that context of use will be and submit that and go through the FDA review for qualification. And this is the challenge with the word validation. Why I say it means different things to different people. Broadly speaking, it encompasses all of this. And so I hope you have a sense that it encompasses an enormous array of activities and things that need to be done. So let's then take a little bit of a deeper dive into what I mean by this context of use. And I'm going to start out by looking at the diagnostic question. So when we're using a biomarker for a diagnostic purpose, we're trying to differentiate someone who has ALS from someone who doesn't have ALS. And we look at these two characteristics, what are known as sensitivity and specificity. Sensitivity is, can we correctly identify those with ALS, what are called true positives, people who really have the disease from people who don't. And specificity is, can we correctly identify those people who don't have ALS, right? So we wanted to correctly categorize people. In this little figure here, we have controls with a low level of the biomarker, and we have patients with ALS with a high level of the biomarker. And here you can see there's a neat line that we could draw between them. Everybody below this level does not have ALS, and everybody above the level does. And we can map that out in this sort of analysis. It's called the receiver operating characteristic curve. And we can ask ourselves how well that biomarker performs in distinguishing people from ALS from people who don't have ALS. Now, we actually have an example of this. We have a diagnostic marker. It's called EMG, or electromyography. This is a test that's used to aid the diagnosis of ALS. But it's not entirely specific for ALS. And so it needs to be looked at in the context in which it's being used. The important thing to say, and I would argue, maybe controversially, and we can have some discussion about it, that diagnostic biomarkers are not the current high unmet need in the ALS world. I think our biggest challenge at the moment is getting somebody who has a suspected diagnosis to somebody who can make that diagnosis. And as you all perhaps know, there's this long diagnostic delay between symptom onset and diagnosis of about a year. And the answer is not biomarkers, it's the social sort of healthcare issue of getting patients to a neurologist who recognizes and understands this disease. Now, by the time that happens, usually the diagnosis is very clear, which is why I don't think a diagnostic marker is what we need. But they may be needed in the future. If we can get people with early disease to seeing neurologists when the disease is much less well established, much less clear, then I think diagnostic markers will become something that we need. So let's then switch and talk about a predictive biomarker. So predictive markers are those that could identify patients with ALS who are most likely to benefit from a particular therapy. So I'm sure it's not lost on this group that ALS may not be a single disease. We know there are many different genetic causes of disease. There are probably many different environmental causes of disease. And so maybe ALS is just a clinical syndrome that has many different causes and many different biologies. And what we would really like to be able to do as a field is know this group of patients have this biology or this cause, and they should be treated in this way. And this other group of patients have a different cause and a different biology and need to be treated in a different way. Now, we do have examples of this. The best example I can give you is that of a genetic mutation. So I'll give you the example of a mutation in the SOD1 gene, one of the more common genetic causes of disease. And when we know somebody has this particular gene that's abnormal causing their disease, we can then say that's the kind of person who's most likely to respond to a gene therapy. Now, that makes obvious sense, and I agree it's a little bit of a banal example, but we don't have those sorts of predictive markers for people with non-genetic forms of ALS. We're very much in the dark. And so I think what we urgently need are these sorts of predictive markers that would tell us this is a patient who may respond to an anti-inflammatory because neuroinflammation is driving their disease. But this other person, neuroinflammation doesn't play a role, and so they're unlikely to respond to that therapy. So developing these sorts of predictive markers would move us closer towards what's sometimes referred to as precision medicine, not necessarily a single treatment for a single patient, but a particular treatment for groups of patients who are most likely to benefit from that particular type of therapy. So what then is a prognostic marker? A prognostic biomarker is one that we can measure when we first see someone with ALS, and it tells us about the future course of disease. How will it unfold? Because we know this disease is enormously variable. With some people, it progresses very quickly, in others, much more slowly. This is a plot of this ALS functional rating scale. This is this quantitative tool we use to quantify how the disease is affecting patients' function. And you can see that the trajectory, how this changes over time, varies enormously from patient to patient. And the problem, of course, when we're doing a clinical trial and we're measuring functional decline on this scale is what happens if this person got active drug, but the person out here got placebo. It would look like the person on active drug was doing worse. And so we need to be able to accommodate or account for this enormous natural variability. So we want to be able to measure the biomarker, and we want it to tell us that the rate of decline might be different between people, say, with a high concentration or a low concentration of the biomarker. Or that the biomarker might tell us something about the future survival or likely survival of somebody with ALS. And this could be enormously helpful, we think, in clinical trials, and that it could take into account or accommodate for this variability and make clinical trials that much more efficient and effective. And we do have examples of these, and I'll show you some data around this, but I think the neurofilaments and this thing that we can measure in the urine, or urinary P75, these do have potential, I think, as prognostic markers. Importantly, if we're validating a prognostic marker, it must add value over and above what we already know from clinical predictors. If I know, for example, that the older age onset people and people of female sex, or people who have bulbar onset disease, if I know that their prognosis is poorer compared to people who don't have those characteristics, and the biomarker doesn't give me any additional information, then it really isn't adding value. So it has to add value to that. And I'll show you some data around that. So then there's this question of pharmacodynamic biomarkers, and that's just a fancy word. Pharmaco is pharmacology, it's drug, and dynamic is change. So it tells us that the biomarker is changing in response to a treatment. And that's the goal of a pharmacodynamic biomarker. So let's look at these plots. So here, for example, there are two different types of these. They're what we might call disease state or static biomarkers, and disease progression or dynamic markers. So here you see in people with ALS and the untreated state, the biomarker is elevated, but very stable over time. In this example, the biomarker is increasing as disease progresses. Now, if we know this is what the natural trajectory is in people who are untreated, and then we give an experimental treatment and the biomarker comes back down to normal, or if it was going up and now it stabilizes, that would give us good reason to believe that that biomarker was doing something. And if we did that in a phase 1 or a phase 2 clinical trial, that would tell us this is the sort of compound that we should move forward into a big phase 3 clinical trial to show that this drug affects not just the biomarker, but also does something important clinically that's meaningful to people with the disease. And so this is currently a problem that we have in phase 2. We often don't know how to interpret phase 2 data. And we don't know which compounds we should move from phase 2 into phase 3. A pharmacodynamic biomarker could be enormously transformative in that regard. And I think we have some potentials here. I think neurofilament is a good example. And there's some promising experience from another motor neuron disease, actually a pediatric motor neuron disease called spinal muscular atrophy. And we now have a treatment for this disease. And we know that when we give it, the levels of neurofilament in the blood go down. And so I think we have a good reason to believe that the same will be true in ALS, although until we have an effective treatment, it's going to be hard to know that for sure. So I'm going to focus this evening on some of the biological fluid biomarker candidates. And I'm doing that because I think they're the most far advanced in many respects and the closest to validation. And some I think probably are validated. And so we'll talk about some of our lead candidates. So we're looking in blood, in serum, and in spinal fluid, looking at the light chain of neurofilament and also the heavy chain. And we won't have a time to talk about urinary P75, maybe on another occasion. And I think the potential contexts of use that these have value as prognostic markers and potentially as pharmacodynamic biomarkers. So if we look at this prognostic utility, and for those not familiar with these graphs, let me just orient you to them. These are what are called survival plots or Kaplan-Meier plots. This is time. Whoops, I need to go back. And this is the survival probability. When we start, everybody is living. And as disease progresses, more and more people succumb to the disease. What you can see here is if you carry a mutation in this gene, C9orf72, survival is worse than if you don't. Likewise, this delta FRS, which is the estimated rate of disease progression when we first see somebody, how quickly has the disease progressed from when symptoms began until now? And the faster they're progressing, not surprisingly, the poorer survival. The slower they're progressing, the better survival. What we see here for neurofilament, and this is in blood, is that high neurofilament at baseline, survival is worse than low. And we see the same effect for the neurofilament heavy chain, although not quite as strong an effect. But you'll notice that each of these graphs are only considering a single factor. And I told you that the biomarker to be valuable, we have to sort of put it into a statistical model to tell us that it's really adding information to what we already know. So we've done this. This is looking at the prognostic utility, and we've looked at age, gender, the C9 repeat expansion, genetic mutation, bulbar onset, limb onset, what your baseline ALS functional rating scale is. He has that estimated rate of disease progression, neurofilament light and neurofilament heavy. And the important thing, what you'll see here, and I've highlighted it, then we put all of this into a statistical model. It really is only two things that jump out that tell us about the future course of disease. How fast you've been progressing until now, so the past predicts the future. And then in addition to that, the level of neurofilament in your blood. And the higher the level, the worse the prognosis, the quicker disease progresses. When we look at this, not just for the slope of functional decline, but also survival, we see exactly the same thing with the addition that the baseline ALS FRSR, how much function you've lost by the time of enrollment in a clinical trial, not surprisingly, is a marker of how far into the disease you are and how much time there is left. So importantly, we think neurofilament and light, not so much heavy, have prognostic value in this way. So what's the relevance of this then to a clinical trial? If we model this out as though we were doing a clinical trial and we adjust for this value of serum neurofilament, disappointingly, well, it does save on sample size. We can do trials that are smaller, but the effect is only modest. So that's a little bit of a gut check. That's an important observation, an important finding. What it means is we should be incorporating neurofilament as a prognostic marker, but it's not going to be game-changing when we use it in this way. But maybe it has value for a different context of use, and we'll come and we'll talk about that. So let's talk about the potential pharmacodynamic utility. Now, importantly, we can't know whether a biomarker has this effect until we have an effective therapy. But we can do some work to get these biomarkers all ready to test out as we have promising therapies that come over the horizon, and we're already doing that. So here's some of the data from SMA. That's that spinal muscular atrophy, that infantile form of a motor neuron disease that's being treated with this drug called nusinersen. And what you can see here is neurofilament light and heavy. Here's light, here's heavy. And with repeated administrations of the drug, this is given into the spinal fluid, you can see the neurofilament levels plummet and come back down to normal. So we have good reason to think the same will be true in ALS. And in fact, there's a little bit of data that that is the case. In Biogen's phase 1, phase 2 study of their gene therapy for ALS, you can see here, I need to get rid of my control bar, whoops. So this here is neurofilament light. I need to wait for my control bar to disappear. And you can see in orange, the group treated with tofersen with the active drug, and you can see in blue, the placebo. And you can see that over time, the neurofilament levels decline in people on active drug compared to remaining fairly stable in people on placebo. And so if we look in people with the fast progressing form of SOD1 ALS, that effect is even more marked. So these are very small numbers, preliminary data, but they do support the idea that neurofilament will be responsive. And so here's what we have found. When we look at neurofilament, this is light and this is heavy and this is in the blood. You can see there's lots of variation between people in their level of neurofilament. But each of these gray lines is neurofilament and how it changes or doesn't change in an individual person. And within each individual person, it's relatively constant. So people establish or develop their own level of neurofilament, and then it remains fairly stable over the course of their disease. So we can use that information. So if we know what the average slope is, and we know in the untreated state what that slope can be, and we're estimating that with some degree of confidence, if we could, for example, give a treatment and we could lower neurofilament further, that would tell us that that wasn't the play of chance, because we don't see that in the untreated state. And that could tell us that the drug is doing something. There's another way we can look at this. I told you that people who progress fast have a higher level of neurofilament, and those who progress slowly have a lower level of neurofilament. And so we can say it would be therapeutically valuable to turn somebody from a fast progressor into a slow progressor. And if we use that difference in neurofilament levels, we can try to begin to answer questions about how neurofilament as a pharmacodynamic marker could be helpful in a clinical trial. And to give you a sense, if we use that first approach I described, we might be able to do a clinical trial with as few as 26 people. If we use that second approach, we might be able to do a clinical trial with as few as 64 people. And in the same data set, if we use the ALS functional rating scale as an outcome, which is commonly used in clinical trials, we would need anywhere from 500 to 1,000 patients to participate in a clinical trial. So this remains a little bit suppositional because we don't, as I say, have an effective therapy. But if this is true, this could be game-changing for how we do phase 2 clinical trials. So let me switch gear a little bit. I'm keeping my eye on the clock and ask whether biomarkers might be able to facilitate a trial in which we want to prevent ALS. So I need to tell you a little bit about PreFALS, or the Pre-Symptomatic Familial ALS Study that we initiated back in 2007. So this is a longitudinal natural history and biomarker study, and we enroll people, healthy people, who carry an ALS gene-causing mutation. And we started with the genetic population, not because we were only interested in genetic ALS, but it was really the only population that we knew to be at a significantly elevated risk for ALS. And we studied people with a whole range of different genetic mutations. They can choose whether or not to learn the results of genetic testing. And the goals were to identify biomarkers of pre-symptomatic disease. So can we measure something before disease becomes apparent? And can we use those markers to predict when ALS is coming? And then how could we use that information to design and implement a disease prevention clinical trial? So just a word to say that the pre and PreFALS, although it stands for pre-symptomatic, it also stands for prediction. We need to predict when disease will develop, because currently in people who carry a genetic risk for disease, we don't know when disease will develop. We want to prepare for a clinical trial to prevent disease, but ultimately the goal is prevention. So when you hear PreFALS and you see pre, think of all of these: pre-symptomatic, predict, prepare, and prevent. So this is what PreFALS looks like. We have a little over 200 people who are enrolled. It's dominated by these two common genetic mutations, SOD and C9. And we've been doing this since 2007, and we have about 1,000 person years of follow-up. And we have unfortunately observed 20 people phenoconvert, move from being unaffected to being affected and having ALS. But those 20 people have taught us an enormous amount. So I want to show you a schema of how we're now thinking about ALS. And we're now thinking about this not just as a clinical syndrome, but really as a biological entity. And this is the tip of the iceberg that we've been seeing for, as long as we've known about ALS, what we'll clinically manifest disease. Symptoms begin, some time passes, and a diagnosis is made. We've long thought that there's a pre-symptomatic period that begins when disease begins, but we haven't had a way to define when disease begins. We've only been able to define when symptoms begin. So as we now develop biomarkers, and we can develop better and better biomarker tools, and they're more and more sensitive, we can move this back and we can begin to use these biomarkers to define when disease actually begins. And so I just want to introduce this term phenoconversion. I've used it already, but it's a term we use to describe the transition from pre-symptomatic to clinically manifest ALS. And we operationally define this as the first emergence of clinical signs or symptoms that in the eyes and the judgment of an experienced ALS neurologist definitely indicate the presence of clinically manifest ALS. And we can get into it in question time if we want to, but it's not quite the same as diagnosis. So here's what we've learned about serum neurofilament in these people who have phenoconverted in pre-phals. So here's time, and this is years since clinical onset of disease, and time 0 is when disease emerged clinically, symptoms began. Here on this axis is the level of the biomarker, the neurofilament, and this gray shading shows us the maximum value of neurofilament that we see in the healthy population. And what you can see is the neurofilament is running along, but in the year prior to developing clinical disease, neurofilament levels go up. They go up and they're above the value that we see in a healthy control population, and they continue to go up soon after symptom onset. This is important. Neurofilament light goes up before phenoconversion, and we can see this as far back as 12 months prior to phenoconversion. And we've seen this in people with rapidly progressive SOD1 mutations. These are mutations that once disease comes over the clinical horizon, disease can progress from beginning to end in about 6 to 12 months. So very aggressive. So if we graph this biomarker data onto this new understanding of disease, we know here's the neurofilament that's stable amongst people who've already been diagnosed, but it's also going up soon after phenoconversion or symptom onset. But we now know it's also going up during this pre-symptomatic period. And this is a phase of axonal degeneration because neurofilament represents degeneration or damage to the axons that are those structural components of the motor nerves that are sick and dying in ALS. So how are these biomarkers relevant to an early intervention or a prevention trial? They have the potential to tell us whom we should treat and when. So who's the whom, at least based on what we know now? It's people with an SOD1 mutation associated with rapidly progressive disease. And the when is when blood neurofilament concentration goes up to a level that exceeds a predetermined threshold that we've set. So this has led us to work with Biogen to design this study called ATLAS. And the goals of ATLAS are to evaluate with a pre-symptomatic treatment with this drug called tofersen, which is something we give intrathecally into the spinal fluid. We do a lumbar puncture, we take off spinal fluid, and it's injected into the spinal fluid. It's an antisense oligonucleotide or an ASO. So it's really a form of gene therapy. And the question is, if we give this to people pre-symptomatically, can we delay the emergence of clinical ALS, or could we even prevent it? And if we can't delay or prevent it, if it still comes, can we slow the functional decline and preserve function? And so we're going to look at the proportion of people who develop clinical disease within 12 months of randomization, which will happen once neurofilament levels go up. So let me explain that a little bit by showing you what this study schema looks like. So the initial part in part A is natural history run-in. This is very much like what we do in pre-phals, and we're screening or we're following people monthly for their neurofilament levels in blood. What we anticipate will happen is that neurofilament levels will go up in some people, and if they're still pre-symptomatic, then they will enter the clinical trial phase of receiving drug or placebo. And we will follow those people until they develop clinically manifest ALS. And at that point, no later than the emergence of clinical disease, they will get open label drug. And our hope is that if people are getting tofersen, we can even delay or prevent the emergence of clinically manifest ALS. We also anticipate there's some people who may develop disease without an elevation in neurofilament, and they'll go into Part D. And this is really a placeholder for now as we wait for the results of the Biogen's VALOR study, which will tell us whether tofersen actually already works in people who are already clinically manifest or symptomatic. And if they are, then this will change from a placebo-controlled period to an open label period. And parts B, C, and D may last up to 2 years. So I'm going to wrap up and just say that I hope I've been able to share with you the idea that I think biomarkers are absolutely critical to therapy development. I think they're critical to development of therapies for people who already have ALS, but also to people who may be at risk for ALS. And we're starting in a small genetic subpopulation of patients, but we think there are enormous learnings that we will achieve that will inform how do we approach this for the broader ALS population. And I think some of the take-home messages are that we need to think hard about this context of use. And I think whenever anybody talks to you about a biomarker, you should ask them how they intend to use it and what its intended use is. And I hope you're thinking about that with respect to your digital biomarkers, because what you want out of them very much depends on the purpose, the intent for which you will use them. And then again, I want to underscore validation. And we all need to think about this beyond discovery. We need to think about validation preclinically and clinically. So there's stuff to do in the lab, but there's also stuff to do with people who are participating in research. And so before I stop, I just want to end with some acknowledgements. And I want to acknowledge all of the people and their families who participated in our studies over many years. I want to recognize and acknowledge the group that I work with at the University of Miami, my colleague Joanne Wu, without whom none of this work would have been possible, as well as our research team, but also the members of our clinic team. Without a robust clinic and providing the care that we do, we know that patients come for care, but also to participate in research. And then my colleagues, Peter Anderson, who's helped with a lot of the genetics that we've done, Christine Stanislaw, our genetic counselor, who's done really most of our genetic counseling, Andrea Malaspina in the UK, with whom we've done a lot of the neurofilament work, and my colleague Lyle Ostro at Hopkins, who's really helped us think clearly about a lot of these biomarker projects, members of the CReATe Consortium, too many to name them all, as well as all of our funding agencies. And so I will stop there and maybe stop sharing so that I can see everybody and we can have some Q&A. So thank you very much.
Thank you.
Hello, Dr. Benatar. My name is Sarah Diaz. I am a Everything ALS team member. I lost my dad in 2018 to bulbar ALS, and I've joined this fight, and now I'm a medical student at the University of Michigan, going to hopefully work in neurology and fight the fight. Our first question comes from our chat, and it is, how would people with ALS benefit from biomarker development?
How can someone with ALS benefit from a biomarker? Is that right?
Yeah, how someone who is currently going through the disease.
So I think they can benefit in different ways. So, let's sort of walk through the different contexts of use. So one of the questions that people ask when they're first diagnosed with ALS is, what does my future hold? How long have I got? What does the future course of this disease look like? So if we had biomarkers that were helpful in predicting that could give people reliable information, I think that would be valuable and helpful, because information is power, and that would be, I think, valuable. More importantly, as somebody with ALS is thinking about, do I want to participate in a trial of drug X or drug Y or drug Z? Wouldn't it be great to say, based on having a predictive biomarker, I think my disease is being driven by this particular biological mechanism. Let's pick neuroinflammation as an example. And so I want to be in a trial of an anti-inflammatory because I think I stand the most to benefit from this. Right now, we don't have that. People participate in trials based on what's available, what they can get into, what their doctor suggests they participate in. So I think it could be enormously empowering. I also think ultimately what we all want for each individual and for the field and for the community is to have effective treatments. And currently, what happens in ALS therapy development is we test drugs in phase 2 and we look at them and we say, they look safe. We can't really tell whether they work. we should do a big, large phase 3 clinical trial with several hundred patients that will cost, I don't know, $100 million and last a few years. I mean, at the end of that, unfortunately, we most often learn that it didn't work. Wouldn't it be great if we could do that in 6 months with 30 patients and decide either it's not working or we think it holds promise and we should really go to the trouble of doing those big clinical trials? So I think these are some of the ways in which I think a biomarker can be helpful. Let me give you another example. When somebody takes an FDA-approved medication, let's say riluzole or edaravone, nobody gets stronger, right? You're taking it on faith because we know that people who in general take these medications progress more slowly than people who don't. But wouldn't it be great to know if it was actually working in you as an individual, that we could say you're taking riluzole, we can see your neurofilament level goes down. Great, it looks like it's working in you. And so I think there are all sorts of potential ways this could be informative and helpful.
Perfect. Thank you.
Long answer to a short question, sorry.
No, it's good. It's absolutely what we need to hear. And the more we can spread the message, the better we'll be able to hopefully get people to go through that.
Got it. Thank you. Hi, Dr. Benatar. My name is Zoe Lalji. My father was diagnosed with ALS in 2017. He's still fighting, so pretty grateful for that. And super grateful for this entire organization. It's part of the reason my family has kept its hopes up for so long. So the next question is, how do you motivate people to engage in this biomarker research?
That's a great question. I think part of it, I think a big part of it is sort of spreading the word. It's education about why biomarkers could be helpful and transformative. You know, very often when I'm talking to patients with ALS, understandably, people want to be in a clinical trial. The unfortunate reality of our history of clinical trials in ALS is that most of these have not yielded drugs that work. And so I think for all the reasons that I gave when I was answering Sarah's question, I think these are the reasons that we have to communicate to people as to why it's important to participate in a biomarker study and to help us develop biomarkers. And I should say that these are not mutually exclusive. I think increasingly clinical trials are incorporating biomarker studies into them, and they're a perfect opportunity to learn about biomarkers. And so I think you can have the best of both worlds, but if you can't get into a clinical trial, you can still contribute to therapy development by helping us to develop these biomarkers.
That answer totally works into our next segue question, and also answers another question in the chat. But how do patient advocacy groups help to facilitate biomarker research? And as a Everything ALS, how can we help to also facilitate other biomarker research and not just our speech study and our own?
Yeah, I mean, I think, again, a big part of it, I think, is about education. It's about spreading the word. But I think what you can also do is educate the community about the rigor that's needed to move biomarkers forward and over the finish line to make them helpful. Because the last thing we want is research that doesn't move the field forward. And so if it's not being done with the rigor that's necessary, then we don't want to do it. And so I think we need to collectively sort of lift ourselves up to say, when we talk about biomarkers and we develop them, let's be crystal clear about how we expect them to be used, what role they might play, and let's make sure that we're engaging in the key studies that can either move them, as I say, up or out, right? If it's going to work, let's move it up. If it's not, let's move it out, stop talking about it, stop studying it, and let's move on to something else. The worst is when we keep talking about something for way too long because we haven't figured out whether we are in love with it or hate it. So I'm in favor of clarity. So I think you can help in spreading that word and sort of really spreading the word about why this is important and encouraging people to participate.
Absolutely.
That's great. Thank you so much. The next question is actually about what you would suggest in eliminating that gap in the diagnostic time for ALS patients, and how you think we should go about it, if not through a biomarker.
Yeah, so we could spend the next half hour talking about that, Zoe. So I'll try to be brief. And I think it's a complicated problem, right? So this has been observed not just in the US, but in parts of Europe, in Canada, really wherever it's been studied. There is this diagnostic delay. And on average, this is about 12 months. We need to understand, why is there a diagnostic delay? So the first thing is that there's some delay when people get symptoms. Sometimes they brush it off and they say, it'll get better, and I'll leave it for a while. And it's only when it doesn't that they go and see their doctor. And then what happens, they go into their primary doctor, and understandably, their primary care doctor is not an ALS expert, not thinking about this. And they say, I think it's carpal tunnel syndrome, or I think it's a pinched nerve in your neck or in your back. You should go and see the orthopedic surgeon. Then you wait to see the orthopedic surgeon. He examines you and says, yep, I think that's right. We should operate on you. So we schedule the surgery. You have the surgery. You don't get better. Right. And you can see that we get into this roundabout of sort of tests and procedures and medical referrals, because I think people perhaps aren't thinking about ALS as a diagnostic possibility. So one of the things we need to do is, and this is where I think you could help enormously, is engage in a public education campaign. So people who think about stroke and heart attack have done this fantastically. If I said to you, somebody was at home and they were watching, I don't know, their favorite sports game on TV, and they developed crushing chest pain and shortness of breath and were sweating, what would you say they had? A heart attack? We'd all be thinking heart attack, right? So in ALS, we need to think, if somebody develops weakness that is painless and is progressing, we need to think about ALS. So painless progressive weakness, think about ALS, and ask to go and see a neurologist or an ALS neurologist. Now, the challenge there is that what that means is we're going to inundate our system. We currently don't have enough ALS neurologists to be seeing all of those people with potential symptoms that may well turn out to be a pinched nerve in the neck or in the back, or a carpal tunnel syndrome. So along with that public education, we need to build capacity of experts to be able to bring that clinical judgment to bear to help figure out, is this ALS or isn't? And then, Zoe, as we start to see people in these earlier stages of disease, that's when we're going to need the biomarkers. And we'll be able to develop those by studying people whom we're seeing early and are struggling with, is this ALS? Is it not ALS? We then need to do those diagnostic biomarker studies then. And just to say it now, because I didn't say it before, if I do a diagnostic biomarker study comparing healthy controls to people who have ALS, I've got a news flash for you. I don't need a biomarker. I can tell just by talking to you that you have ALS or you're a healthy control. And so any biomarker that only differentiates those populations isn't going to help us. We need the biomarkers in the space where the ALS expert is genuinely unclear. So I don't know, a long answer, I could say more, but I hope that sort of gets at the question.
Yes, thank you so much. That was right on point.
Our next question is for the people that are currently here, wondering if they are able to participate in any of the studies that you presented today, and also if they're in another clinical trial. Is it possible to participate in biomarker research while being on a medication, even if it is placebo?
Yes and yes. So we have many biomarker studies that are ongoing. Some are occurring multi-center around the country, but they do require being able to get to a clinical site. But we're not the only group doing biomarker studies. There are many others. And so I'd say the best biomarker study, not the best, but I'm being a little bit glib, but the best one is the one that you can actually participate in. So talk to your local clinic and ALS center about those opportunities. But if you live in South Florida, if you live in Kansas, if you live in Pennsylvania, we have other sites where people could go if they would like to participate. What was the second part of your question, Sarah? Ask again.
If you're in
in a clinical trial. Yeah. Absolutely. So, there are very few clinical trials that would preclude you from also participating in a biomarker study. And in a biomarker study, if you're also in a clinical trial, we want to know it, we want to document it, because if that drug turns out to work, right, that's going to affect the interpretation of the biomarker data. So we need to know that. So you can still participate, but we have to keep track of it and we have to know the details. And then when you eventually learn if you were on placebo or active drug, we need to know that, because it will aid the interpretation of those biomarker data down the line.
Wonderful. Thank you.
So the next question is regarding whether you had any information on the TDPBP studies.
It's a big question. So TRDBP is TAR DNA binding protein. It's the gene that encodes for this protein called TDP43. And TDP43 is the major pathological hallmark of ALS. And so I'm not sure what specific studies somebody is asking about. I think there are many groups that are studying TDP43. I don't know, can you get clarity on that question, Zoe, and I can try to answer?
Yeah, I'll ask the chat.
Are you able to kind of talk a little bit about the P75? I know you presented a bit about it in your presentation, but just kind of speak on what it is for those who don't know and why it might be important.
Yeah, so that's a great question. Let me back up a little bit and say that when we're looking at biological fluids, one of the reasons we're very interested in spinal fluid is because the spinal fluid bathes the brain and the spinal cord. So it's the closest thing we can get to actually studying the brain and the spinal cord. So it's helpful to study spinal fluid. The challenge is not everybody wants to undergo a spinal tap. Not everybody wants to undergo a spinal tap regularly. And so it's a much harder fluid to access to be able to study. So further away from the brain and spinal cord than spinal fluid is blood. So blood's a useful place to study, but blood is a complex fluid. It's got lots and lots of different proteins in it that are doing all sorts of different things. And so it's a hard sort of material to work with. So that leads us to think about urine. Urine's pretty straightforward. There's not a lot of stuff in it, at least there shouldn't be in health, but it makes it simpler to get at. And it's also pretty simple to collect. Not many people would mind peeing in a cup to give us a urine sample. You could probably do it every day if you wanted to. It's easy to do. It's very non-invasive. So it's an attractive matrix or fluid for us to test. And P75 is something discovered by a colleague of mine in Australia, Dr. Mary Louise Rogers, and we've done some work with her to say, how could this be useful? But P75 is a marker of axonal degeneration like neurofilament. But unlike neurofilament, which we don't measure so well in the urine, we can measure P75 in the urine. And unlike neurofilament, which once it goes up in ALS and then stays stable, we think P75 continues to go up. So we think it may have different value as a biomarker in telling us about the progression of disease. And maybe we don't know it will be differentially responsible to treatments. And so it's another potential pharmacodynamic biomarker. But again, we don't know that yet, but we're trying to ready it to help us answer that question as soon as we have some therapy that's effective, even for a small subset of the ALS population.
That's great. Thank you. So the next question is, what areas of focus do you think would be most impactful in discovering new biomarkers?
So I have my own biases. I'll try to express them, but also get beyond them. So, I think that the biological fluids, which I focused on tonight, hold the most promise. And I say that not because I don't think there's value in the imaging and the physiology. I think there is, but they're not as well developed. And I'll tell you why. So the key thing about the neurofilament, for example, as a biofluid measure, is that we can measure it with precision at an individual patient level. I don't have to look at groups of patients and compare groups to groups. I can look at you and your neurofilament and say something about you. And I think there's a power in that sort of individual utilization of a biomarker that there isn't when I have to say, yeah, I'm doing an MRI, but I've got to do it in 30 people and then look at the average result to be able to say something. And I think that may change over time. If I look at the field of multiple sclerosis, for example, imaging biomarkers have been transformative for the field of developing therapies for multiple sclerosis. So we could get them to that point, perhaps for ALS, they're not there yet. So my own bias is that the biofluid markers are closest, but we also want to be spending time moving some of these other markers that might teach us different things forward and either up or out. Can we get them ready and better and ready to use, or we should move them out? So I think there's value, but it's not where I would sort of be putting my penny down right now.
That answer kind of directly goes into our next question about neurofilaments. In regards to, like you said in your presentation, it also shows for SMA and other diseases. How can it be used specifically as an ALS biomarker if it does impact more than just one neurologic disease?
Right. So there's so much more in that question, Sarah. That's just fantastic. So what you're touching on is the lack of specificity for neurofilament. So when neurofilament goes up, That could be ALS, it could be a pinched nerve, it could be multiple sclerosis, it could be a stroke, it could be a traumatic brain injury, you name it. All sorts of things can send neurofilament up. But that's precisely why I think neurofilament is not going to be a diagnostic marker. Because when it's up, it's not going to say ALS, it could be any one of those other things. But I think the lack of specificity doesn't matter when we switch from thinking about it as a diagnostic marker to instead use it for a different purpose. So if we know your neurofilament is up and now we can bring it down with a therapy, that's telling us something different, and it doesn't matter that it's not specific. So that's the essence of what this context of use is all about, that what properties we want the marker to have really depend on how we're going to use it. And I worry we've been throwing the baby out with the bathwater because we haven't been sort of teasing those conceptual elements apart.
Thanks. So the next question is actually a clarification from the TDPBP question. The person that asked it was actually just wondering whether there are biomarkers for that gene.
So, I mean, the only biomarker for the gene, right, is looking at the gene itself. But more than the gene, this is a sort of another holy grail in the ALS world, is that we know that with the exception of patients who have an SOD1 mutation or a mutation in the FUS gene, just about everybody with ALS, when they pass and we look at their brain and spinal cord, they have TDP43 pathology in the brain. The problem is we can't measure that in life. We don't have a good test. So look at, for example, and there's controversy around this, but look at, for example, the FDA's recent approval of the Alzheimer's drug, aducanumab. They approved that on the basis of a biomarker, that the drug lowered amyloid in the brain. Amyloid to Alzheimer's is like TDP43 to ALS. So if we had a biomarker of TDP43 and we could show we were clearing it from the brain, man, that would be incredible in terms of helping us to develop therapies. So that's a high priority area of research for us as a field.
Awesome, thank you.
In terms of children of people with ALS, how are they able to get involved in clinical trials, whether or not they've been identified with an ALS gene?
So I think there are two broad populations within that group that you've described. So you could be the child of somebody with ALS, where that person with ALS is the only person in the family, or you could be the child of somebody with ALS, where ALS runs in the family. I know we're running low on time, but which of those were you asking about? And I'll try to address my comments there.
So if you have, I guess, for the person who doesn't have ALS or known ALS and wants to participate because a family member, a direct family member, like a parent may have it. I don't know.
So I think it sort of depends. So if you don't have a family history of ALS, meaning there's only one person with disease, not multiple people, and if there isn't a genetic cause of your disease, you could sign up for a biomarker study and be a control. We need controls. We need people who don't have disease to help inform the interpretation of those data. If, on the other hand, you're the child of somebody who's got a genetic form of ALS and there's a family history, then you could participate in a study like Pre-Fals of people who are at genetic risk, where we can try to better understand when disease is coming and what we might be able to do to prevent it. So I think there's a nuance there, depending on sort of which of those groups you fit into. There might be different opportunities.
Thank you. Yes.
Okay, so this is actually going to be our last question. Could you speak about how what the process is in signing up for one of these biomarker studies, and also how participation looks like, what it would look like to be a participant?
Yeah, so it's complicated because there are many different studies and each study is a little bit different. So, I mean, I can certainly send to you, and you can send out to your membership information about websites, email addresses, phone numbers that people can contact to see whether there's an opportunity for them. The best way to learn about what it entails is we do have on websites, publicly available information, but talk to one of the research coordinators or research assistants and they'll tell you about it. They'll tell you this is a longitudinal study. You need to come back every 3 months. We're going to collect blood. We're going to collect urine. If you're willing, we want to collect spinal fluid too. If you're willing, we're also going to do an MRI. And so different studies might entail different procedures. And so that's a key part of the informed consent process, that it's our responsibility to communicate that. But we have a lot of this information on our various websites if people want to go and look it up and then come sort of armed with that information to ask those questions.
Wonderful. Well, Dr. Benatar, thank you so much for your time today. This has been thoroughly informative. And I don't know if you're seeing the chat, but everybody has been just all about this presentation. I know I'm going to have to rewatch and re-listen and research myself so I can truly absorb everything that we discussed tonight. But thank you again for your time. And we hope to get some big progress here soon. And if there's anything that we can do specifically, please let us know. We'll definitely take that information and send it out to our team. Now we go to an open forum so everybody can kind of talk. Feel free to stay if you'd like to, but we understand it is late on the East Coast. I'm an East Coaster too, so it's, you know, a little past time, but we really again appreciate you coming and joining us this evening.
No problem. I'm going to drop off, but let me just extend my thanks to you. This is just great work that you're doing, and I appreciate the opportunity to talk with you, to share thoughts, to hear your questions. And so thank you for all of what you're doing and for your interest and for your future and current participation in research to help us move forward. So thank you.
Absolutely. Thank
You.
Bye, everyone.
Thanks, Dr. Benatar. Thank you.
Sure. Thanks, Lisa.
Bye for now.
you. All right.
Bye.